Hello everyone. So today we are going to learn about human blood substitutes. I'm Sanskar Datta from SBC Bangalore, doing 4th SEM. So what are human blood substitutes basically? So they are basically a fluid which improves the transportation of oxygen inside the system, inside your body. Human blood substitutes. With the increasing demand for blood transfusions, the development of blood substitutes has become essential in the medical field. In this presentation, we will explore the history types, development challenges, and advancements of human blood substitutes. The two major types of blood substitutes are volume expanders, which include solutions such as saline that are used to replace lost plasma volume, and oxygen therapeutics, which are agents designed to reduce oxygen normally carried by hemoglobin in red blood cells. And the picture you can see it's blood transfusion is happening. So basically, what are human blood substitutes? And there are three types: hemoglobin based oxygen carriers, which can be said as HBOCs. HBOCs are developed by extracting hemoglobin from human or animal blood, purifying and chemically modifying it to increase its stability and oxygen carrying capacity. Next is perfluorocarbon based oxygen carriers, PFCs. So they are synthetic molecules that do not bind directly to oxygen but act as oxygen carriers through dissolution of oxygen in the liquid. So the third one is synthetic oxygen carriers as SOCs. So these are entirely synthetic molecules that can bind and carry oxygen in the same way as hemoglobin. So why are they needed basically? So in case you have blood shortage or anything due to the limited supply of human blood, substitutes can be used and is an alternative. So just to donate blood and all, it's basically used. And transportation challenges: blood substitutes can be transported and stored more easily than natural blood, reducing the risk of spoilage and wastage. The third is compatibility: blood substitutes are compatible with all blood types. So basically it means that for all blood it's applicable for all blood group. So the history of blood substitutes. So in 1600s, the first animal blood infusion was carried out. 1697, Richard Lower, an English physician, transfused the first human blood. In 1929, ADP, adenosine diphosphate, was discovered, leading to the development of blood storage techniques. In 1980s, the development of blood substitutes started as a response to the AIDS epidemic and the need for a safer blood supply. So this is the history of blood substitutes. So types of blood substitutes. Hemoglobin based oxygen carriers is the first one, which is HBOCs. These blood substitutes are developed using residual hemoglobin and can carry oxygen like natural blood. Second is perfluorocarbon based oxygen carriers, PFCs, contain no hemoglobin and carry oxygen through dissolved oxygen in the liquid phase. Next is synthetic oxygen carriers, SOCs. They are entirely synthetic molecules that can bind and carry oxygen same way as hemoglobin. So the development challenges, regulatory hurdles. Blood substitutes face strict regulations and long approval processes before they can be used in medical settings. Safety concerns. Blood substitutes have to pass rigorous safety checks before they can be used, as they can cause severe side effects such as allergic reactions or high blood pressure. efficiency issues. Blood substitutes have to be carefully tested to ensure that they perform their required function effectively and they do not compromise patient health in any way. So there, this is basically the safety concerns and also how to what to do before you doing the blood substitutes. Advancement in blood substitute research. So in 2015, researchers from University of Essex found a way to use spider's silk to create blood clots and plug wounds. Essex is basically in England, so it happened in England. Scientists in 2020, scientists successfully developed artificial red blood cells using 3D printing technology. 2009, nanotech researchers successfully created a blood substitute that can mimic the real thing by using nanoparticles. So this is the process for artificial blood substitutes. First is skin or blood cells are taken from a human donor from the universal blood group O negative, O positive. Induced pluripotent stem cells are then cells are genetically rewound into a stem cell-like state, creating induced pluripotent stem iPS cells. iPS cells are cultured for 1 month in a chemical environment designed to nudge them towards becoming red blood cells. They become matured. The matured red blood cells, which make up 40 to 50% of the total cells, are separated in a centrifuge. Then the red blood cells are ready to be used in a transfusion in the patient's trial. So what and all we learned from today? The benefits, challenges, and the future. The benefits for the blood substitutes can help to reduce the risk of blood shortages, transportation difficulties, and compatibility. Challenges you might face: the development of blood substitutes faces regulatory, safety, and efficiency issues that require careful consideration and testing. Future would be the recent advancement in research has shown promising results, and the future of blood substitutes is looking bright. So this is what we learned in this PPT about human blood substitutes, and thank you.